Hello all. So today we are talking about Chapter 6, Part 2, Protein, Metabolism, and Function. So what happens to protein in the human body? Dietary protein consists of long, complex chains of amino acids in the digestive process. Enzymes, proteases, and the stomach and small intestine break the complex protein down into polypeptides and then into individual amino acids. The amino acids are absorbed through the wall of the small intestine, pass into the blood, and then pass to the liver via the portal vein. The digestion of protein takes several hours, but once the amino acids enter the body, they are cleared within 5 to 10 minutes. There is a constant interchange of amino acids among the blood, the liver, and the body tissues. The liver is a critical center in amino acid metabolism. It is continuously synthesizing a balanced amino acid mixture for the diverse protein requirements of the body. These amino acids are secreted into the blood and carried as free amino acids or as plasma proteins such as albumin. All these functions consume energy, and the thermic effect, or TEF, is greater for protein than for carbohydrate and fat. Whether or not this plays an important role in weight control is discussed in Chapter 10. The most important metabolic fate of the amino acids is the formation of specific proteins, including the structural proteins such as muscle tissue and the functional proteins such as enzymes. Body cells obtain amino acids from the blood, and the genetic apparatus in the cell nucleus directs the synthesis of proteins specific to the cell needs. The body cells may also use some of the nitrogen from the amino acids to form non-protein nitrogen compounds, such as creatine. For example, the muscle cells will form contractile proteins as well as the enzymes and creatine phosphate necessary for energy production. The body cells will use only the amount of amino acids necessary to meet their protein needs. They cannot store excess amino acids to any significant amount, although the protein formed may be catabolized to release amino acids back to the blood. Because the human body does not have a mechanism to store excess nitrogen, it cannot store amino acids per se. Through the process of deamination or breakdown, the amino group containing the nitrogen is removed from the amino acid, leaving a carbon substrate known as alpha keto acid. The excess nitrogen must be excreted from the body. In essence, the liver forms ammonia. From the excess nitrogen, the ammonia is converted into urea, which passes into the blood and is eventually eliminated by the kidneys into the urine. This is just a summary of protein digestion and absorption. And you can see the portal protein digestion to the enzyme pepsin and the stomach acid. Proteins broken down. Further protein digestion by enzymes is released by the pancreas. And final digestion of the protein into amino acids, mostly inside the cells of the intestine. And then amino acids are absorbed into the portal vein and transported to the liver. From there, they enter the general bloodstream, and our little dietary protein is present in the feces. Typically, it goes into the urea. Here, stomach acid and enzymes contribute to protein digestion. The enzymatic protein digestion begins in the stomach and ends in the absorptive cells of the small intestine, where the last peptides are broken down into a single amino acid. The alpha keto acid that is released may have several fates. For one, this carbon substrate may be oxidized for the release of energy. For another, it may accept another amino group and be reconstituted to an amino acid. It also may be channeled into the metabolic pathways of carbohydrate and fat. The liver is the main organ where this conversion occurs. In essence, some of the amino acids are said to be glucogenic amino acids, that is, glucose forming. At various stages of the energy transformations within the liver, the glucogenic amino acids may be converted into glucose. As noted in Chapter 4, this process is called gluconeogenesis. The ketogenic amino acids are metabolized in the liver to acetyl-CoA, which may be used for energy production via the Krebs cycle or converted to fat. The glucose and fat produced may be transported to other parts of the body to be used. Thus, although excess protein cannot be stored as amino acids in the body, the energy content is not wasted, for it is converted to either carbohydrate or fat. In essence, some of the amino acids are said to be glucogenic amino acids, that is, glucose forming. At various stages of the energy transformations within the liver, the glucogenic amino acids may be converted to glucose. As noted in Chapter 4, this process is called gluconeogenesis. The ketogenic amino acids are metabolized in the liver to acetyl-CoA, which may be used for energy production via the Krebs cycle or converted to fat. The glucose and fat produced may be transported to other parts of the body to be used. Thus, although excess protein cannot be stored as amino acids in the body, the energy content is not wasted, for it is converted to either carbohydrate or fat. Protein turnover represents the process by which all body proteins are being continuously broken down and resynthesized, and it is an ongoing process. The National Academy of Sciences indicates that about 250 grams of body protein turns over daily in an adult, or about half a pound of body mass. Figure 6.5 represents a summary of the fates of protein in human metabolism following the digestion of dietary proteins. One of the major functions of the amino acids is the synthesis of body tissues, enzymes, hormones, and antibodies. However, protein is constantly being degraded by the liver. The excess nitrogen is excreted as urea, while the carbon residue may be converted into carbohydrate or fat or used to produce energy. Can protein be formed from carbohydrate and fats? Yes, but with some major limitations. Protein has one essential element, nitrogen, which is not possessed by either carbohydrate or fat. However, if the body has an excess of amino acids, the liver may be able to use the nitrogen-containing amino groups from this excess amino acids and combine them with alpha keto acids derived from either carbohydrate or fat metabolism. A key alpha keto acid from carbohydrate is pyruvic acid, while fat yields acetoacetic acid. The net result is the formation of the body of some of the non-essential amino acids using carbohydrates and fats as part of the building materials. Keep in mind that nitrogen must be present for this to occur, and its source is dietary protein. What are the major functions of protein in human nutrition? Dietary protein may be utilized to serve all three major functions of food. Through the action of the individual amino acids, protein serves as a structural basis for the vast majority of body tissues and is essential for regulating metabolism, can be used as an energy source. In one way or another, protein is involved in almost all body functions. Its individual roles are beyond the scope of this text, so the following discussion represents just some of its major functions of importance to health and fitness. Table 6.5 highlights the major functions of protein in the body. Protein is the main nutrient used in the formation of all body tissues. The role is extremely important in periods of rapid growth, such as childhood and adolescence. Athletes who attempt to gain muscle tissue also need an adequate dietary supply of protein to create a positive protein balance. Certain amino acids, such as the branched chain amino acids, leucine, isoleucine, and valine, constitute a significant amount of muscle tissue. Protein is critical in the regulation of human metabolism. It is used in the formation of all enzymes, many hormones, and other compounds that control body functions. Insulin, hemoglobin, and the oxidative enzymes in the mitochondria are all proteins that have important roles in regulating metabolism during exercise. Other metabolic roles of protein include the maintenance of water balance and acid-base balance, regulation of the blood clotting process, prevention of infection, and development of immunity to disease. Proteins also serve as carriers for nutrients in the blood, such as the free fatty acids and the lipoproteins, and help transport nutrients into the blood's body cells. Although protein is not a major energy source for humans at rest, it can serve as a function under several conditions. In nutritional energy essential balance, the priority use of dietary protein is to promote synthesis of body proteins, essential for optimal structure and function. However, as noted previously, excess dietary protein may be deaminated and used for energy and may be converted to carbohydrate or fat and then enter metabolic pathways for energy production or storage. During periods of starvation or semi-starvation, adequate amounts of dietary or endogenous carbohydrates and dietary fats may not be available. Both dietary protein and the body protein stores are used for energy purposes in such a situation. But because energy production takes precedence over tissue building and metabolism, hence, if the active individual desires to maintain lean body mass, it is essential to have not only adequate protein intake, but also sufficient carbohydrate calories in the diet to provide a protein sparing effect. In other words, carbohydrate calories will be used for energy production, thus sparing utilization of protein as an energy source and allowing it to be used for its more important structural and metabolic functions. So protein, of course, is used as an energy source. During rest, it's about 5% of total daily expenditure. And with endurance training, it may reduce protein oxidation at rest, increasing the amount of energy derived from fat. Protein uses for energy during exercise as well. Exercise and protein losses do occur. So protein metabolism during recovery from exercise and effective training on protein metabolism is important. The protein needs of athletes and prudent protein recommendations for athletes. So are proteins used for energy during exercise? Protein has always been considered one of the main staples of an athlete's diet. In this section, we discuss several topics regarding protein and exercise, including its use as an energy source, possible avenues for protein loss and protein metabolism during recovery, and dietary requirements and recommendations for strength and endurance athletes. Are proteins used for energy during exercise? The average individual consumes about 10% or more of daily energy intake from protein. For individuals in protein and energy balance, this protein intake must be balanced through energy expenditure and other body losses. In general, scientists suggest that about 5% of daily protein intake may be used directly for energy. Protein in excess of tissue needs may be converted to carbohydrate or fat, which may also be used for energy. Protein may also be lost from the body in various ways, such as creatine and urea excreted in the urine and sloughed body cells. However, given its nutritional importance, researchers have attempted to determine the effect of exercise on protein balance and needs. Scientists have used a variety of techniques to study protein metabolism during exercise because urea is a byproduct of protein metabolism. Its concentration in the urine, blood, and sweat has been analyzed. Also, the presence in the urine of a marker for muscle protein breakdown known as 3-methylhistidine, a modified amino acid, has been studied to evaluate protein metabolism. The nitrogen balance technique consists of precisely measuring nitrogen intake and excretion to determine whether the individual is in positive or negative protein balance. Finally, labeled isotopes of amino acids have been ingested or injected to study their metabolic fate during exercise, not only in the whole body but also in isolated muscle groups. Using these techniques, investigators have evaluated the use of protein during both resistance exercise and aerobic endurance exercise. Although both types of exercise affect protein metabolism, the use of protein as an energy source appears to be more prevalent in aerobic exercise, particularly when prolonged. In a review, Rennie and Timpton noted that resistance exercise causes little change in amino acid oxidation, but probably depresses protein synthesis and elevates breakdown acutely. In this regard, Evans suggested that the eccentric muscle contraction associated with resistance training may produce ultrastructural damage that may stimulate this protein turnover. However, Wolff noted that factors regulating muscle protein breakdown in human subjects are complex and interactive, and proposed that muscle protein breakdown is paradoxically elevated in the anabolic state following resistance exercise. Thus, while it appears that resistance training does not increase protein or amino acid oxidation, it may provoke muscle tissue breakdown. Portmans has noted that although protein may be used to produce significant amounts of ATP in the muscle during aerobic endurance exercise, the rate of production is much slower than with carbohydrate and fat, the preferred fuels. On the other hand, Rennie and Timpton noted that sustained dynamic exercise stimulates amino acid oxidation, mainly by activating an enzyme, BCAA dehydrogenase, or BCAAD, that oxidases BCAA and increases ammonia production in proportion to exercise intensity. If the exercise is intense enough, there is a net loss of muscle protein as a result of decreased protein synthesis, increased catabolism, or both. Some of the amino acids are oxidized as fuel, and the rest provide substrates for gluconeogenesis. In this regard, prolonged exercise may be comparable to a state of starvation. As the endurance athlete depletes the endogenous carbohydrate stores, the body catabolizes some of its protein for energy or eventual conversion to glucose. Protein catabolism has been shown to increase significantly even when muscle glycogen is depleted by only about 35 to 55%. Remember, catabolism is a breakdown of the protein. Metabolism, in general, a brief session of exercise lowers the rate of protein synthesis and speeds protein breakdown. The exact mechanism of protein metabolism during exercise have not been determined. Though several mechanisms have been proposed, Parkhouse has reported that exercise, particularly exercise to exhaustion, activates specific proteolytic enzymes in the muscle that degrade the myofibrillar protein. Fitz and Metzger found elevated levels of proteolytic enzymes in fatigue muscle. In a review, wagon makers indicated that 6 amino acids, BCAA, asparagine, aspartate, and glutamate, may be metabolized in the muscle, providing the nitrogen needed for the synthesis of ammonia, alanine, and glutamine. During exercise, particularly prolonged aerobic exercise, Graham and MacLean noted significant muscle efflux of ammonia, glutamine, and alanine. These 3 products carry excess nitrogen from the muscle to other parts of the body, most notably the liver for recycling or conversion to urea. Ammonia, a nitrogen byproduct of protein catabolism, is an indicator of increased muscle amino acid breakdown. Although no underlying mechanism has been identified, increasing levels of ammonia in the body have been associated with fatigue, somewhat comparable to the accumulation of lactic acid. One theory is that increased amino levels in the muscle may impair oxidative processes, thus decreasing energy production. While another theory suggests that increased plasma ammonia may impair brain functions and induce central fatigue. Because ammonia is formed in the muscle from the amino group, removal of the amino group by alanine and glutamine may help decrease the production of ammonia and delay the onset of fatigue. Glutamine release from the muscle also increases during exercise, and it is an important fuel for various cells in the body, particularly those in the immune system. As we shall see later, glutamine supplementation has been studied as a means to enhance immune function in athletes. Although a number of amino acids may be used as energy substrate during exercise, the major research effort has focused on the fate of leucine. Wagon makers noted that the increase in BCAA oxidation during prolonged exercise seems to be specific for leucine only. In essence, the amino group of leucine catabolism eventually combines with pyruvate in the muscle cell to form alanine and leaves the residual alpha keto acid. The alpha keto acid may enter the Krebs cycle and be used for energy production. The alanine is released into the bloodstream and transported to the liver, where it is converted into glucose. The glucose may then be released into the blood to be used by the central nervous system and may eventually find its way to the contracting muscle to be used as an energy source. Alanine appears to be the most important means of transporting the amino group to the liver for excretion as urea. The overall process involves gluconeogenesis, known as the glucose-alanine cycle, and is depicted graphically in Figure 6.6. Some investigators have noted that during the cycle, during the latter part of endurance exercise, the blood levels of alanine increase, presumably because more is released from the muscle. However, the estimated glucose production approximates only 4 grams per hour, which might make a limited contribution in mild intensity exercise, but is possibly insignificant during high intensity exercise, when carbohydrate use may approximate 3 grams of more per minute. Additionally, several investigators have reported an increased release of branched chain amino acids, or BCAAs, from the liver during endurance exercise, with subsequent uptake by the muscle cells. This figure 6.6 is the glucose-alanine cycle. The alanine may be produced in the muscle tissue from the breakdown of other amino acids, most notably leucine. The alanine is then released into the blood, travels to the liver for eventual conversion to glucose through the process of gluconeogenesis. Protein or amino acids can be utilized during exercise to provide energy directly in the muscle and via glucose produced in the liver, particularly when the body stores of glycogen and glucose are low. In earlier research, Lemon reported that in the latter stages of prolonged endurance exercise, protein could contribute up to 15% of the total energy cost. However, less protein would be used for energy during the early stages of prolonged exercise when carbohydrate is adequate. Thus, Gibala suggested that the contribution of amino acid oxidation to total energy expenditure is negligible during short-term intense exercise and accounts for 3 to 5% of the total energy production during prolonged exercise. In his review, Tarnopolsky cited similar figures, indicating that protein oxidation could account for 1 to 6% of the energy cost of aerobic exercise. Tarnopolsky also noted that women oxidize less protein compared to men and show lower leucine oxidation during aerobic exercise. It is important to note how dietary carbohydrate influences protein as an energy source during exercise. A low carbohydrate diet leading to a decreased muscle glycogen level will lead to increased dependence on protein as an energy source. However, adequate carbohydrate intake before and during prolonged exercise will help reduce the use of body protein for this purpose. Because the presence of adequate muscle glycogen appears to inhibit enzymes that catabolize muscle protein, scientists from the University of Maastricht in the Netherlands noted that high carbohydrate diets may have a protein-sparing effect for endurance athletes. Although the available evidence suggests that metabolism of protein and its use as an energy source are increased during exercise, the magnitude of its contribution may depend on a variety of factors such as intensity and duration of exercise and the availability of other fuels such as carbohydrates, either as stored muscle glycogen or consumed during exercise. Does exercise increase protein losses in other ways? Other than loss of protein from oxidation, exercise has been shown to increase protein losses from body in several other ways. Exercise may cause an elevated level of protein in the urine, a condition known as proteinuria. This condition has been observed following competition in a wide variety of sports, including running, football, basketball, and handball. Research suggests that the greater the intensity of the exercise, such as a high-intensity 400-meter sprint versus a lower-intensity 3,000-meter run, the greater the loss of protein in the urine. Prolonged aerobic exercise, such as the triathlon, has also increased urinary protein loss. Yaguchi and others suggested that heavy, prolonged exercise, such as a triathlon, may induce some transient kidney damage in some individuals. Such damage may explain the findings of Portmans, who reported a decreased reabsorption of protein in the kidney tubules following intense exercise. However, the total amount of protein lost in this manner appears to be rather negligible, amounting to less than 3 grams per day. Protein also may be lost in the sweat. Several investigators have reported the presence of both amino acids and proteins in exercise-induced sweat, with both sweat rate and sweat nitrogen losses increasing with greater exercise intensities. Again, the losses are relatively minor, on the order of 1 gram per liter of sweat in adult males. This avenue could account for 2 to 4 grams of protein in an endurance athlete training in a warm environment. As Shelby noted in Chapter 8, prolonged intense exercise may also increase gastrointestinal losses of iron, which may be bound to blood proteins. Again, however, these protein losses would be relatively minor. So what effect does exercise training have upon protein metabolism? Rennie noted that protein metabolism may also become more efficient as a result of training, noting that the response of muscle protein turnover to habitual exercise is comparable to other metabolic changes in the muscle associated with exercise training. Although an initial bout of exercise may markedly elevate protein breakdown and synthesis in an untrained individual, the effect would be much less in one who has trained habitually. Tarnopolsky indicated that training induces a decreased activity in BCAAD, the enzyme that oxidizes BCAAs, when exercising at a standardized workload of the same absolute intensity. As mentioned, protein synthesis appears to predominate in the muscles during recovery. With training or repeated bouts of exercise on a regular basis, changes in the muscle structure and function are additive. Numerous studies have found that after resistance or endurance training, protein balance becomes positive. Trained individuals during rest have been shown to experience a preferential oxidation of fat and a sparing of protein as measured by leucine metabolism and the respiratory quotient, or RQ. The specific exercise task apparently stimulates the DNA in the muscle cell nucleus to increase the synthesis of protein, and the type of protein that is synthesized is specific to the type of exercise. Aerobic exercise stimulates synthesis of mitochondria and oxidative enzymes, which are composed of protein, are necessary for energy production in the oxygen system. Resistance training promotes synthesis of the contractile muscle proteins. These adaptations are the key factors underlying improved performance. So these figures are adaptations in body tissues following exercise training that are specific to the type of exercise. The exercise stressor influences the genes to initiate the formation of protein synthesis through the transcription, transfer, and translation processes. The transcription process provides the template, the messenger RNA, or mRNA, for the specific protein. The transfer RNA process moves specific amino acids to the ribosome. The translation process by the ribosomes, or ribosomal RNA, forms the protein. Aerobic exercise training, that's A, will lead to an increase in serum hemoglobin levels, which transport oxygen to the muscle cells. Resistance training will lead to an increase in skeletal muscle protein. That's the second there. Additionally, other than its beneficial effects in increasing structural and functional protein important to resistance or aerobic endurance training, training may influence protein metabolism in other ways to help prevent premature fatigue. You may recall from previous chapters that there is substantial research to support the conclusion that aerobic endurance training improves the ability of the muscle cell to use both carbohydrate and fat as energy sources during exercise. Although extensive evidence is not available, in a review, Graham and others noted that following endurance training, enzymes in the muscles appear to develop the potential for increased capacity for oxidation of leucine and the other BCAAs, which are an abundant source of energy in the muscles. Thus, endurance training may increase the capacity of the muscle to derive energy from protein in a fashion similar to the increased utilization of fat. Another possible means to spare the use of carbohydrates, such as blood glucose, to help protect the main energy source for the brain and muscle glycogen. Although these changes do not appear to spare the use of muscle protein, they would appear to spare the use of carbohydrate, and thus may help prevent fatigue when carbohydrate levels are decreased during exercise. Additionally, when exercising at a standardized workload before and after training may also decrease the production or accumulation of ammonia. Extrapolating from animal research, some investigators theorize that instead of forming ammonia, the nitrogen is incorporated into other amino acids, such as alanine, for transportation from the muscle to the liver. Theoretically, reduced plasma ammonia levels may be associated with less fatigue. Training properly may also help prevent muscle injury associated with eccentric exercise. Research shows that physically trained individuals, when compared to untrained individuals, do not experience as much muscle tissue damage during prolonged eccentric exercise tasks. However, specificity of training is also important in individuals preparing for a race with a significant downhill component, such as the Boston Marathon, should gradually incorporate exercise intensities of eccentric exercise into their training. So does exercise increase the need for dietary protein? In their joint position stand, the American College of Sports Medicine, the Academy of Nutrition and Dietetics, and Dietitians of Canada concluded that protein requirements are higher in very active individuals and suggested that endurance and resistance athletes need 1.2 to 1.7 grams per kilogram of body weight. In a similar vein, the International Society of Sports Nutrition, in a position stand developed by Campbell, and others concluded that protein intake of 1.4 to 2 grams per kilogram body weight daily may improve body adaptations to exercise training. Conversely, in its dietary reference intakes for protein, the National Academy of Sciences concluded that in view of the lack of compelling evidence to the contrary, no additional dietary protein is suggested for healthy adults undertaking resistance or endurance exercise. As Stuart Phillips, a protein metabolism expert, points out, these two divergent points of view are likely caused by defining protein needs or requirements with body nitrogen balance, as opposed to defining optimum protein intake based on maintenance or gain of muscle, bone, and connective tissues. Skeletal muscle, bone, tendon, and ligament protein turnover, which can be 50 times slower than gut or plasma proteins, are likely not well represented in a short-term nitrogen balance study. This section focuses on the optimum protein intake for those individuals seeking to increase or maintain the functional capacity of skeletal muscle connective tissue, such as athletes. Our ability to maintain or increase skeletal muscle mass is based on our ability to properly balance two processes: muscle protein synthesis, or MPS, and muscle protein breakdown, or MPB. Following the ingestion of a high protein meal from either food or protein supplement, there is a period of time when blood levels of amino acids increase dramatically. This amino acidemia stimulates MPS, or muscle protein synthesis, while the resulting increase in blood insulin, hyperinsulinemia, inhibits muscle protein breakdown. In this scenario, which fluctuates throughout a normal day, a positive protein balance occurs when muscle protein synthesis is greater than muscle protein breakdown, which can stimulate muscle hypertrophy and increase strength. As described by DeVries and Phillips, two factors that greatly influence MPS and MPB are resistance training and energy intake. Following a resistance exercise workout, both MPS and MPB are increased, with muscle protein balance achieved when protein is consumed following the exercise. When energy intake is restricted, which occurs as athletes as well as the general population, MPS decreases, which can cause a negative protein balance and subsequently a decrease in muscle mass. As discussed later in the chapter, the addition of protein to the diet may help to prevent negative protein balance and muscle loss during times of energy restriction. A common question by those attempting to increase muscle mass and strength with resistance training is whether or not additional dietary protein will cause a further increase in muscle hypertrophy and strength. The answer to this is unequivocally yes. A recent meta-analysis by Cermak and colleagues supports the need for additional protein to promote the best adaptations to resistance training. In young adults, the ingestion of additional protein of greater than 1.2 grams of protein per kilogram of body weight per day during resistance training 6 to 24 weeks caused a 50% greater increase in muscle fiber cross-sectional area and 20% greater increase in maximal strength than resistance training and placebo ingestion. And this is just a recommendation. Some exercise scientists recommend an optimal intake of about 1.5 to 1.8 grams per kilogram of body weight per day. It is important to reinforce the viewpoint that carbohydrate is the main energy source for endurance-type athletes. Besides its efficiency as a metabolic fuel during exercise, carbohydrate also provides a potent protein-sparing effect. Consuming sufficient carbohydrate will decrease reliance on protein during aerobic endurance exercise, reduce the formation of ammonia, and better maintain normal protein status in the body. Nevertheless, additional dietary protein has been recommended for endurance athletes because they may utilize protein as an energy source during exercise and may need protein to synthesize oxidative enzymes in mitochondria. Athletes involved in prolonged, intermittent, high-intensity sports such as soccer, field or ice hockey, lacrosse, and basketball may also fall into this category. Additionally, extra protein has been recommended to prevent development of anemia during the early stages of training. In brief, sports anemia is thought to occur because the body uses dietary protein for these aforementioned purposes at the expense of hemoglobin formation, leading to anemia. Sports anemia has been associated with iron nurture and will be discussed in more detail in Chapter 8. Moore and colleagues recently reviewed the dietary protein needs of endurance athletes and noted that muscle remodeling is needed to break down old and damaged proteins and synthesize new proteins. Acute endurance exercise and chronic endurance exercise cause an increase in MPS, or muscle protein synthesis, and this adaptive response to endurance training can be enhanced by providing additional dietary protein. As with strength-trained individuals, various investigators have also estimated the protein requirement of endurance-trained individuals. In a review based on protein balance studies, Lemon recommends that individuals involved in vigorous aerobic endurance exercise consume 1.1 to 1.4 kilograms per kilogram per day. Slightly higher amounts, about 1.4 to 1.7 grams per kilogram of body weight, have been recommended for individuals involved in intermittent high-intensity sports because these include a balance of endurance and power activities. The following mathematical presentations are possible scenarios for various athletes who wish to maintain or increase protein balance. The protein needs are based upon the RDA, a rough estimate of the amount of protein used during strenuous exercise, and additional protein needs for the individual who want to gain weight. Let us look at the young resistance-trained athlete who wants to gain body weight, preferably in the form of muscle tissue, through a weight training program. The protein RDA for an adult male is 0.80 grams per kilogram. At moderate activity levels, the average 70 kilogram male would be in protein balance with about 56 grams per day. However, according to the suggested upper recommendation of about 1.7 grams per kilogram, he would need about 119 grams per day if involved in strenuous training program. Is this a reasonable amount? 1 pound of muscle tissue is equal to 454 grams, and its composition is approximately 70% water, 7% lipids, and 22% muscle tissue. Hence, 1 pound of muscle contains about 100 grams of protein. If the desired weight gain is 1 pound of lean body mass per week, a reasonable goal, then this young male would need to assimilate an additional 14 grams of protein per day to supply the amount in 1 pound of muscle tissue. A gain of 2 pounds per week, although probably more difficult to accomplish, would require the assimilation of 28 additional grams of protein per day. Let us be liberal and estimate an additional 22 grams of protein per day to cover losses due to exercise. In summary, assuming that a portion of those protein needs are not covered by the safety margin incorporated in the RDA, this young athlete would need approximately 106 grams of protein per day to gain 2 pounds of lean body tissue per week, or about 1.5 grams of protein per kilogram of body weight. This value falls within the recommended range for resistance-trained athletes. So, ideally, you want to obtain 15% or more of daily energy intake from protein. As do most investigators, Peter Lemon indicates that it is possible to get these recommended amounts from natural food sources in the daily diet. For example, the average caloric intake for a moderately active young male averages 2,500 to 3,000 calories. This caloric intake may be increased through physical training as more calories are expended. Thus, caloric intake would be increased to approximately 35 to 4,000 calories. It is important to note that adequate energy intake, primarily in the form of carbohydrates, will improve protein balance. In essence, an increased energy intake appears to decrease protein requirements somewhat. If the protein portion of the dietary calories average 12%, a general recommended level of protein intake, then the intake of protein would approximate 1.5 to 1.7 per kilogram body weight, which parallels the amounts estimated in the examples cited previously. Currently, the protein content of the average American diet is 12 to 16%. And Wahlberg-Rankin noted that most athletes consume this much or more in their daily diets. Consuming a diet with a protein content of 15% could provide a value of 2 grams or more per kilogram of body weight, and other surveys among strength-type athletes indicate that they obtain this amount. These values approach or exceed the higher amounts recommended by some investigators for individuals in training. The calculations are presented in Table 6.6. So this is a protein intake for a wrestler, a college wrestler of 132 pounds or 60 kilograms. RDA is 48 grams of protein per day, so a diet of 1,600 calories to maintain his body weight. A diet with 12% protein will provide the RDA. If you see, it would equal 48 grams of protein. A diet with 20% protein will provide about 1.7 grams of protein per kilogram per day of weight, which is near the recommendation for strength athletes. So, So with that, he would be getting in 80 grams of protein per day. We also need to consume protein, preferably with carbohydrate, before and after workouts. Eating strategies may also be an important consideration, particularly timing of protein intake. Most research has focused on amino acid or protein ingestion following exercise, but some studies have also studied the effect of pre-exercise protein intake. The feedings usually occurred immediately before or within an hour of exercise. Many studies have used protein hydrolysate, a high-protein dietary supplement containing a solution of amino acids and peptides prepared from protein by hydrolysis. Wolf noted that the protein supplement should contain all the essential amino acids, and many studies also added carbohydrates. Protein intake before exercise. In separate reviews, Tipton and Wolf both noted research studies suggesting that the ingestion of free amino acids plus carbohydrates before exercise results in a superior anabolic or building response to exercise than if ingested after exercise. Lemon indicates that the practice of consuming nutrients prior to exercise may be beneficial by providing fuel and or minimizing catabolic processes. However, in his review, Tipton noted that the difference in anabolic response between pre-exercise and post-exercise ingestion of protein is not apparent. In 2008, the International Society of Sports Nutrition published its position stand on nutrient timing. As part of this position stand, Kirksick and others stated that the optimal pre-exercise meal is dependent upon several factors, including exercise duration and fitness level. General guidelines they suggest are ingestion of 1 to 2 grams of carbohydrate per kilogram of body weight and 0.15 or 0.25 grams of protein per kilogram body weight in the 3 to 4 hours prior to competition. Perhaps the two most frequently asked questions by those interested in adding protein to their diet are, what is the best time to eat protein, and is one protein better than another? These questions are often in reference to which time and what type of protein will cause the greatest increase in muscle protein synthesis, muscle hypertrophy, and strength. As reviewed by Van Loon, many proteins including whey, casein, soy, casein hydrolysate, egg, whole milk, and fat-free milk have been shown to improve post-exercise protein balance or muscle protein synthesis. However, the goal of an athlete is maximal or optimal MPS. There are a wide variety of protein supplements available for athletes and other consumers to choose from, including whey, casein, egg, soy, and collagen. During the past few years, experts in protein metabolism from the laboratories of Stuart Phillips, Kevin Tipton, and Luc Van Loon have published research studies that have helped to answer these two questions about protein. The two most commonly consumed protein supplements are the two proteins in milk, casein and whey. Data from the Phillips group have demonstrated that whey protein stimulates MPS more than casein or soy protein, both at risk and post-exercise. It makes sense that whey protein causes the largest increase in MPS because whey is rapidly digested, exits the stomach quickly, and results in a large increase in blood amino acids. Whey is a complete protein containing all the essential amino acids, and whey is high in the amino acid leucine with about 3 grams of leucine per 25 grams of whey protein. Conversely, soy protein has lower levels of essential amino acids, branched chain amino acids, and leucine. Also, soy protein is less bioavailable when whey or casein are and when consumed, and is more likely to be oxidized or involved with organ protein catabolism and urea synthesis. Casein, although it is digested, and absorbed more slowly than soy, has higher levels of essential amino acids, branched chain amino acids, and leucine than soy. Collagen is an incomplete protein, is low in essential amino acids, branched chain amino acids, and leucine, and is a poor choice for a protein supplement. The existence of a post-exercise anabolic window and importance of post-exercise protein ingestion in general has recently been called into question by Schoenfeld, an expert in muscle hypertrophy. In a meta-analysis, Schoenfeld showed a beneficial effect of post-exercise protein consumption on muscle hypertrophy, but this effect was not evident when total protein intake was considered. He contends that the benefit of post-exercise protein ingestion was not necessarily due to timing, but simply to greater protein consumption. Also, Aragon and Schoenfeld have pointed out that much of the post-exercise protein consumption research is conducted with volunteers in a fasted state. That is unlikely to be the case with real athletes. They suggest pre- or post-exercise protein ingestion of about 0.4 to 0.5 grams of protein per kilogram of lean body mass as general guideline to create the optimal anabolic effect. Practically speaking, an inexpensive, nutrient-dense food such as fat-free milk or cottage cheese would be an excellent choice for high-quality, high-essential amino acid, high-leucine food. However, athletes often seek the optimal adaptive training response and may choose to consume a protein supplement at certain times during the day. Additionally, if an athlete is attempting to optimize muscular adaptations or training as well as decrease body fat, a post-exercise leucine-enriched, low-dose whey protein supplement might be the logical choice. Finally, the constraints of sport can influence dietary protein intake behaviors. Some athletes compete multiple times per day, which would necessitate a fast digesting protein, while others travel a great distance to a training facility or competition and may not have access to a refrigerator. Few research would argue that protein supplements are necessary. Rather, it is optimal to consume a high-quality protein several times per day to support positive protein balance. This can be achieved with high-protein foods, protein supplements, or a combination of the two. However, in any discussion of protein supplements versus high-protein foods, it is worth noting that humans shouldn't focus on eating individual nutrients, but rather whole foods that supply a variety of essential nutrients. For example, a protein supplement may contain high amounts of essential amino acids. A food such as milk may also contain high amounts of essential amino acids as well as important vitamins such as vitamin D, important minerals such as calcium, important macronutrients that support human performance such as carbohydrate, and much more. Similarly, a food such as lean beef contains high concentrations of essential amino acids, but also amounts of the minerals iron and zinc. As demonstrated in Table 6.8, protein supplements generally cost more than high-protein foods. Although there is nothing inherently wrong with protein supplements, they should be treated as just that, supplements to a healthy diet. Few researchers would argue that protein supplements are necessary. Rather, it is optimal to consume a high-quality protein several times per day to support positive protein balance. This can be achieved with high-protein foods, protein supplements, or a combination of the two. However, in any discussion of protein supplements versus high-protein foods, it is worth noting that humans shouldn't focus on eating individual nutrients, but rather whole foods that supply a variety of essential nutrients. A protein supplement that may contain, and we've already said that. Based on available research findings, the ratio of carbohydrates to protein requires additional investigation. As concluded by the International Society of Sports Nutrition, it is in its position stand on nutrient timing. And this position stand, Kursick et al. Conclude that the approach often used is to consume a supplement containing a ratio of 3 to 4 grams of carbohydrate per gram of protein within 30 minutes following exercise, although commercial protein, carbohydrate, sports drinks, and supplements are available. Phillips and others recommend a high-quality protein food source such as dairy protein, eggs, and lean meat, which provide abundance of essential amino acids. In their review, Rennie and others note that a solution of mixed amino acids will increase human muscle protein synthesis to about the same extent as complete meals. A turkey breast sandwich on whole wheat bread consumed with a glass of chocolate skim milk provides a good balance of protein and carbohydrates. Carp and Elliott, along with their associates, indicated that milk, including chocolate milk, may be an effective recovery drink for athletes and provide an alternative to supplements. It should be noted that although these dietary practices may provide the nutrients necessary for an anabolic response in the muscle, Gabala indicates it remains to be determined if the acute effects of supplementation eventually lead to greater gains in muscle mass following habitual training. In a study involving a 10-week resistance training program, Cribb and Hayes found that consuming a supplement containing protein, creatine, and glucose immediately before and after training as compared to consuming the same supplement in the morning and evening resulted in greater increases in lean body mass, muscle cross-section area, and muscular strength. Although this study would appear to support anabolic effects of protein and carbohydrate intake immediately before and after exercise training, the supplement also contained creatine, which, as noted later in this chapter, may it be by itself increase muscle mass and strength. Most investigators in the area indicate the more research is needed to determine the optimal combination of protein and timing for the various types of exercise training. So a key point is the major function of dietary protein is to build and repair tissues and to synthesize hormones, enzymes, and other body compounds essential in human metabolism, but it also may be used as a significant source of energy under certain conditions. And that concludes our discussion today. We'll be picking back up with the part 3 of chapter 6. I look forward to seeing you then. Thanks. Have a great day.